Okay. Can this volume go up a little bit? Okay. Oh, that's much better. Thank you. Thank you. Welcome, everyone. Welcome to the November Public Observatory Night that is all astrobiology themed, which is the search for extraterrestrial life. Some of you may recognize me from the Public Observatory Nights. If you haven't, I'll quickly introduce myself. My name is Philippe. I'm the outreach manager at the Harvard College Observatory. I organize these public nights. If you want to know more, please take one of my business cards. It's on the table at the back. Send me an email, and I can give you more information about the Observatory Nights. So I normally pick themes of these nights based on something that's happening in the news or something that's happening up in the sky. But for this time, none of that is true. For anyone who knows me well, in a past life before I did outreach, I was a researcher of astrobiology. So I'm an ex-astrobiologist. And so astrobiology is very close to my heart. And so that's why I picked this theme. It was just inevitable that I'll eventually pick this theme for one of these nights. So a quick little rundown for the schedule. There's obviously not going to be any roof observations. Sadly, not in this weather. But we're going to have two fantastic talks by our guest speakers that I will introduce in a moment. And then after that, after the talks, we aim to finish about 7 o'clock. And what we're going to do is we're going to have an exhibition of some historic artifacts on the back table over there. And one of our curators, Lisa Bravata, who's right there, she's going to take the lead for explaining and introducing some of these prized possessions that we have here. In addition to that, I'm also going to be offering a tour of one of the most beautiful telescopes that I've had the privilege of looking at. We have this enormous telescope here that was built in 1847. And it's called the Great Refractor, and it's truly majestic. So if you're interested at the end of the talks, then follow me and I'll give you a brief 20-minute tour of the Great Refractor. All right. So very, very quickly, I couldn't do an astrobiology themed night without giving a quick 2-minute fun fact about what I used to do in my past life. So I'm going to be 2 minutes just explaining what I think is one of the coolest concepts in science, which is microbial entombment, a possible pathway detecting alien life. Okay, so 2 minutes, I promise. What we're looking at right now is a spider encased in amber. Now, I have this weird obsession with amber. I love amber. I'm half Polish. I spend a lot of time on the Baltic Sea, where there's a lot of amber you can find on the coast. And the reason why I think amber is so awesome is because it has the ability to preserve biology in exquisite detail, also in 3 dimension. This spider, take a guess how old this spider is. Any guesses? Yeah, what have you got?
Spider's probably around a little bit, I'm guessing 27 million years.
That's a very, very good guess. This spider is about 250 million years old. So it's about 250 million years old. And you can still see the hairs on its legs. And we can even analyze its last meal in its gut. So why am I talking about this spiders? Because when I was doing astrobiology, I was really jealous of the potential or preservation of biology that amber can offer. But anyone who knows the environment of space, we're not expecting to find amber in extraterrestrial environments like on Mars, because in order to have amber, you need trees, because amber is fossilized tree sap. And so it turns out there is something that is similar to amber, and that is salt. When I say salt, I'm talking about the salt that you're aware of. So it's table salt, also referred to as sodium chloride. In chemistry, salt can also be like a whole family of different minerals. And the reason why salt is so fascinating is because when salt grows, so salt is a crystal, and when it grows in a salty environment, if there's some form of microbiology present in this environment, then sometimes the microbes can get trapped inside the salt crystals. And when this happens, the microbes can be preserved for millions and millions of years. In fact, one of the craziest stories ever, in the year 2000, there was a paper that was published where some researchers found some bacteria inside a block of salt from the Alps. And this bacteria was dated to 250 million years old which is not the craziest part. The craziest part is the fact that this bacteria was still alive. This was bacteria that was not sitting there as a deceased, kind of shriveled up remnant of a living thing. It was actually a viable organism. This paper, take it with a pinch of salt, pun intended, because it is deeply controversial. And so a lot of people kind of dismiss it. But even if it is not true, there are countless papers that talk about this phenomenon of microbial entombment, of microbes being stuck inside of these salts and being preserved for millions of years. This is a photograph that I took in my lab. This is a teeny tiny piece of salt that has near 1 million microbes trapped inside of it. And soon, this exact piece of salt, which is the size of a pinhead, will be on exhibition in a museum in Scotland, which is very exciting. And linking this all back to the search for extraterrestrial life, turns out that some places in the solar system, like Mars, is very, very salty. This is a photograph taken by Curiosity, and this right here, these are salt veins crisscrossing this landscape. And one day soon, we are going to return some of these samples from Mars to Earth. And hopefully, my hypothesis that the first ever detection of alien life will come true, and it'll be found. We might found alien life inside one of these Martian salt veins. So that is my fun fact for you all. If you want to know more, then come find me at the end of the talks, and we can talk about some salty aliens. But for now, I'm going to introduce my first speaker of the night. We have Dr. Jenny Calahan. Jenny is a postdoctoral fellow at the Center for Astrophysics, Harvard and Smithsonian, and studies the environments in which planets form. She primarily works with models simulating environments that we have observed with the largest radio and infrared telescopes, including the James Webb Space Telescope. She uses models and observations together to better understand the physics and chemistry evolution in planet-forming disks. She completed her PhD from the University of Michigan in 2023 and her undergraduate degrees from the University of Arizona. Jenny Calahan's title for tonight is How to Build an Earth and Other Habitable Planets. Please, enormous round of applause for Jenny Calahan.
Thank you so much for that introduction, and thank you all for coming today. I'm excited to tell you, a little bit about not only the search for extraterrestrial life, but also how we get life and how we understand it. So it's a small task, of course, but we have to start somewhere, right? So we're going to start at the very beginning. I'm going to start with the Big Bang. Long, long ago, around 13.6, 13.7 billion years ago, we had nothing, and then all of a sudden we had something. And that something was mostly just hydrogen and helium, the most simple atoms in the periodic table and in our universe today. That's not much to work with. We can't really make life out of just hydrogen and helium. So there had to be some sort of evolution throughout the universe to get where we are today. So this is what humans are made of versus what we started with in the very beginning. Let me see if this works. Yes. So you can see in the very beginning, vast majority of the universe, just hydrogen, helium, trace amounts of other things, but nothing crazy. While humans over here are mostly made of oxygen, carbon, maybe a little bit of hydrogen in our water, and then some really important other trace metals like sulfur and phosphorus and iron. None of that was found at time zero. So how do we get to just the simplest building blocks, which are atoms beyond hydrogen and helium? Well, we have to form them inside of stars. So the first stars were these really weird objects, nothing like the stars that we see in our night sky now. They're really massive, only made out of helium and hydrogen, probably very short-lived. None of them are around today, but they created the first signs of carbon, nitrogen, and oxygen, which are fundamental to life. Those went on to form the next population of stars, still very high mass, low metallicity objects. They created some more carbon, nitrogen, oxygen, slowly polluting the universe with these metals. And now we can also introduce interesting things like supernovae and high energy phenomena, like the dead cores of stars merging together to create even heavier elements such as sulfur, phosphorus, silicon, iron, and anything beyond that, which is all fundamental to life. So, okay, pretty easy. A few million to billion years have passed, and now we have all the atoms we need to build life. How do we go from atoms to molecules? So these supernovae will enrich their local environment, and that environment will look something like this, a molecular cloud somewhere in space made of gas and, importantly, dust. So gas is really just the loose assortment of atoms. And then dust is usually made of iron, silicate, these heavy metals, these refractory elements. And this is actually where the chemistry is happening, is on these tiny little dust grains. And so this is a nice little animation. If we zoom into a dust cloud and into a single grain of dust, you can see that atoms are able to fall onto this dust grain, and they are now on a 2D surface. So this allows for chemistry to happen much more efficiently than it would happen in a 3D gas phase. And so this is an example of water forming. So you see your hydrogen, your oxygen finding each other and forming water on the surface of this grain. And this will continue to happen until you build a full sheet of ice, and you can build other molecules as well. But this is basically how we go from loose atoms into molecules into even more complicated things. A lot of it is happening kind of in the vastness of outer space on these dust grains. So now you can see we're fast forwarding, we're building up a ton of water. We can build other molecules here as well, and it continues from then on out. So we have our molecular clouds where we can form molecules. That's great. How do we actually get that to a planet or a place where we might have other life out there? Well, before we get to a planet, we have to build a star. So we have a protostar that'll look something like this. This is an image from the JWST telescope. So you can see there's a lot going on. The star is right in the center. We're not really seeing the star, but we're seeing how the star affects the environment in this image. So you can see that there's a lot of things happening. There are jets, there are winds. And so it's a chaotic event for sure. If you zoom in even closer to where the protostar is, you also form what we call a protoplanetary disk. This is where the planets are actually going to form. This is made of gas and dust as well. And this is actually what I study. And then we can form planets inside of this disk, and we actually have direct images now of young baby planets forming inside of these protoplanetary disks, which is very cool. Eventually that gas dissipates and you're left with just a dusty disk of debris, so that will eventually form into planets and comets and asteroids. And then you finally have your planetary systems, including our system and all the exoplanets that we've discovered, which we've discovered well over 5,000. And this is where we're going to be looking for life. But if we want to understand how to look for life, what we should really be looking for, we want to understand how life can form on these environments. We need to understand the chemistry of these origins of life along every stage of this path. So I'm just going to highlight some cool science that's coming out of each of these fields. So in molecular clouds, we've been staring at them for a long time with nice radio telescopes. And if you stare at them long enough, you can get a signal from different molecules. Every molecule has a very unique fingerprint that we can detect with the radio telescope. So we've cataloged nearly 100 molecules in these cold, dense molecular clouds that don't have any stars near them, that are just out there in the cold vastness of space. We already have the ingredients for the origins of life. If we could just scoop up the icy gunk and put it on a planet, that's all we need. But there are many other stages here. So that's good to know that we're starting out with complexity right from the very beginning. We can see what's happening actually on the ice grains, which is pretty magical with a telescope like JWST. So if you have a bright star behind the environment of your protostar, you can actually, with these absorption features, basically catalog what is in the ice phase. And so we can test our theories of chemistry on these ice grains, which is normally very hard to probe. Within protoplanetary disks, there's a lot of cool science happening, but we know that chemistry is somehow processed between the protostar phase before it gets into planets in these protoplanetary disks. How much chemistry is inherited versus totally restarted and processed in the disk is a very active area of research. And then we can look at these disks that are actually hosting planets and look at the chemistry within these like tiny little baby planets. Water is a very interesting molecule that we think is pretty fundamental to life, at least as we know it. So we are able to detect water within these systems in the gas, and we can see how that evolves over time, where the reservoir is in a disk and how it gets transferred to a planet. This dusty phase, for example, in our solar system, this is where we think we got the bulk of our water, where we got our oceans from. The Earth is not a particularly water-rich planet, so it probably did not directly inherit the water in the gas phase in the disk. It probably got it from comets. So that is a critical stage. And then very excitingly, in the field of exoplanets, we can now detect atmospheric compositions of exoplanets, which is wild. You can see a planet from like the other side, or, many, many parsecs, many, many light years away. And we can see that there's water. We can see that there's carbon monoxide, carbon dioxide, and other things. We can see if there are clouds, what the clouds are made of. It's a pretty exciting time to be in this field right now. So I'm just going to zoom in. So far in this talk, I've kind of focused on the delivery of the origins of life. And we see that there's a lot of interesting chemistry in outer space. And it's fairly straightforward to deliver those ingredients to a planet. There are a lot of open questions there, but we can get those ingredients there. At some point, I've totally skipped over how life is created. I'm not going to talk about that in this talk. Javi's going to talk about it in the next one. I'm going to actually jump forward to talk about how we will detect life on other planets. Yes. Okay, so before I do that, I do want us all on the same page of like, okay, where should we be looking for life? What are the key ingredients you need to have a planet, a moon, whatever, that has life? So here I've just found 5 things that I think are pretty essential for life. There might be other things, but these are maybe the top 5 things that you need. So first of all, it's helpful if you have a surface. This sounds kind of straightforward. This is kind of similar to what I was saying with dust chemistry. So you either need a planet or a moon. You need something that confines all of the ingredients you need to form life in one space. So basically, we have all those complex molecules in molecular clouds, but there's not a way to kind of contain all those ingredients together to form life. So we're going to be looking at planets, moons, etc. Easy enough. Another thing we need is just a solvent. So this is something that allows chemistry to happen fairly quickly. We know that water is a great solvent. It allows for chemistry to happen. It's not going to impact the chemistry too much. So water seems like a good thing to look for, liquid water. We need some sort of energy source. And this is not exactly my field. Javi is going to talk more about this, but it seems like in the early stages, not only do you need those ingredients, not only do you need a place for those ingredients to find each other, but you also need some sort of energy source to kick off the critical chemistry to form life. And so in our own Earth, we think this could have come from a variety of things. So we have these deep sea thermal vents that create a lot of energy and have all the ingredients for life. Maybe life started there. Or there are these primordial lakes, very similar to soda lakes today on Earth, that have all the primordial gunk, and they are exposed to energy as well. So life could have started there. Another interesting thing about the Earth is that in the early days, the Hadean Earth, the Earth was much more UV rich. We didn't have ozone. And so that is another energy source that can kickstart life. And then, yeah, get excited for the next talk. We'll talk about that. And then after that, once you've kind of kickstarted life, what do you need? Well, I think you actually need just the right amount of hostility on a planet in order to form life that is resilient and that will last a long time. So examples on the Earth, we went through many very extreme ice ages, basically. So our Earth went through the stage called the snowball Earth at some point, but life survived and persisted and became very strong and resilient. We've also had late stage meteorites that wiped out the dinosaurs, and life continued to survive past that. So it seems like you kind of need that to have long lasting life. And once you have long lasting life, you can find multicellular life, animals, plants, humans, etc. Once you have that kind of life, it's not very useful to have an energy source. It's not very useful to have this kind of UV bombarding your planet. And so Earth eventually evolved to be a very sustainable place for life, a very calm place for life. And so we have a nice atmosphere. We have a nice magnetosphere that's protecting us from radiation from the interstellar medium. There are also a lot of things that just seem really great about Earth. Our tilt isn't too extreme. Our orbit around the sun is not too elliptical. So temperature gradients throughout the seasons and from day to night aren't changing too much. And that's just really great for life. So this is kind of what we're looking for when we're looking for potential life out there. Okay, how do we actually detect life? What are the mechanisms? We can look for biosignatures. So this is basically, we think metabolism is a key thing for something that is alive. It should have some sort of metabolism. All that means is that you have your environment, you're taking resources in, you do something with those resources, and then you excrete them out. Those excretions are biosignatures. So we're looking for molecules that wouldn't have an abiotic origin. So we can look for that. I'm going to talk a little bit more about biosignatures, but I wanted to mention a few other things that astronomers are doing to look for life. One is looking for technosignatures. So this is basically being sensitive to advanced life that might be out there relatively close to us. So us humans, we've been emitting radio signals for over 100 years now. And if you multiply that time since the radio signals were originated times the speed of light, you'll get a distance, and that's basically the radius of our radio sphere of influence. And there are actually multiple stars and multiple exoplanets that are within our sphere of influence. And so if there are any advanced civilizations that have the technology to be sensitive to the radio signals that we're putting out there, they could totally hear us. And perhaps we have the same opportunity here on Earth. So people are trying to listen for alien life that is out there. It's a very difficult thing to do, but people are doing it. And then I think kind of the most exciting thing, which Philippe actually talked about, is this direct detection direction. So this can be, you're looking for biosignatures or ways that life has impacted its environment. So this could be microfossils, this could be little microorganisms stuck in salt. And I think this is kind of the easiest way to convince people that we found life is through some sort of direct detection. But biosignatures are cool. And we're looking for biosignatures on exoplanets. So I just wanted to highlight two examples of some of the most popular biosignatures that have been used and put out there recently. So this first one is called dimethyl sulfide. It is a product that's produced by marine microbes on Earth, and it has no known abiotic origin. So basically, it can't just be rock byproduct. Seems like you need some sort of life to produce dimethyl sulfide. And we have a detection of dimethyl sulfide in an exoplanet in K218b. This is an AI-generated image of it, but maybe it looks like that. And this is what the JWST spectra looks like. So all of these dots are detections from the telescope. And then the blue line is actually a model that is fit to these sparse data points. And the authors of this paper say that this right here is a signature of dimethyl sulfide. It's kind of a weak signature, but they put it out there, and I think it's close. However, we now have detected within this year dimethyl sulfide on a comet as well. And so that's not great for this being a biosignature, because we know that there's not life on this comet. This is just a cold, dusty ice ball that's been exposed to radiation for millions of years. There's no way there's life here. So there must be some sort of abiotic origin for dimethyl sulfide. Another example, famous one, is phosphine, this pH3 product. Very similar. It can be produced by life forms. And we would not expect it to exist for a long time on any terrestrial planet. We do see it in gas giants, but we shouldn't expect to see it on, say, Mercury, Venus, or Mars. We do see it here on Earth, but there's life here, turns out. But there was a detection of phosphine on Venus that was presented a few years ago. This is the magical detection right here. It's very, very faint. It's very hard to pull out this kind of signature. And when they put out this and they were like, hey, there's a biosignature of life on Venus, they propose that there are microbes in the cloud deck of Venus. So Venus, if you go to the surface, is very inhospitable to life. We have sent missions there, we have sent spacecraft there, and they have melted on impact, basically. So we know that there's not life on the surface, but if you go up into the clouds, into the atmosphere of Venus, there are pockets where the temperatures and the pressures are more friendly for life. I don't know how life would form there, but if it was there, it could be there, and it could produce phosphine. However, since this was a pretty major discovery, people really looked at this with a microscope, and since then, this observation has kind of been debunked. But they're going to continue looking for it. And fun fact, even though this was debunked in the time between publishing and debunking, a Venus mission was approved. So we're going to get a mission to Venus regardless, and we'll be learning much more about Venus. Okay, so what are the most promising signs for life? I haven't given you any hope so far about finding life out there, but I think it is promising. I think the most likely place to find it is within our own backyard. And not literally, I mean that in an astronomical context, of course. I mean, we should be looking in our solar system. So one of the most exciting options is looking at a moon around Jupiter called Europa. This is a promising sign for life because it has a subsurface ocean. So there we go. We have our water. to create life. It has an energy source. We have the sun. It also has Jupiter pulling on it. So it has tides. It has this kind of tidal force that produces enough energy that could form life. We would expect to find microbes here, if anything. Nothing too complicated, but that would be really cool. And we actually have a mission that just got launched this year that is going to Europa. It's not going to land on the surface, but we'll be able to learn a lot more about Europa and see if it is still a likely candidate for finding life. Titan is a very similar story. It's a little bit less likely because it's much colder than Europa, a little bit more farther out there. But we also think that this has a subsurface ocean of some sort. There could be ancient signs of life on Mars. So Mars today is not very friendly for life. There's not a lot of water. The water that is there is super salty, but Mars looked a lot different a few million years ago. It looked more like the Earth. It had these massive oceans. And so perhaps life was kickstarted there, stuck around a little bit, and then dissipated. But there could be ancient signs for that life on Mars. I think that'd be really, really cool and quite possible that we could find that. And then I still want to highlight that there maybe could be microbes in the cloud deck of Venus. I think this is the least likely option, but we're going back to Venus. We're going to get more information, and then we can make a judgment call. So just going back to the very beginning, we are made of all the stuff in the universe. We came from practically nothing. That became a lot of something. A lot of complex chemistry is just out there in outer space. And somehow that all coalesced into one tiny little speck of dust that is our Earth. I think there's life out there. Absolutely. Will we see it and find it in our lifetime? Who's to say? It's a big old universe. It is hard to get all of these critical things together in one location to form life. The universe is huge, our galaxy is huge, and we are only really able to probe a tiny, tiny part of that galaxy. So it's going to be really hard to find this evidence. Will we understand how life forms? I'm going to give you the very optimistic answer of maybe. And that's all I have. So I'm happy to take any questions. Thanks for listening.
Thank you, Jenny, for an amazing, amazing talk. We have a couple of minutes for questions. Does anyone have any questions for Jenny? Any questions? Up there. Was there a question? Yeah, go.
They exploded the galaxy.
How can we explore the galaxy?
How can we explode the galaxy?
Explode the galaxy? I don't know. I think that would be very, very difficult / impossible. But that's a fun idea. Yeah. Sure, why not?
Where did Mars water go? And should we be? Where did Mars water go? And should we be concerned about bringing samples back from Mars?
Oh, interesting. Okay, that's a good, you're testing my knowledge a little bit. So you might know better than me, actually. I think Mars doesn't have a very strong magnetic field. And so it doesn't have a good protection, basically. So I think the water just evaporated away. It just left. I don't think we should be worried about bringing samples back from Mars, especially I don't think we should be worried about I don't know, loose aliens accidentally getting loose on Earth. I trust the scientists. They're pretty good at keeping things intact, and I don't think there's going to be a live life in those samples, certainly. I am more afraid of us contaminating Mars. We have a lot of life teeming absolutely everywhere. And so we could bring that to Mars and then accidentally discover our own life on Mars. I fear that could happen, but I'm not too worried about the samples coming back here.
Hi, Jenny. Thank you for the great talk.
Thank you.
If you had all the money you need for one space mission or a telescope, where would you look for life at?
I would want to stay within our own solar system, which I think is kind of controversial. If we were talking to any other exoplanet scientists, they would say they want a massive IR optical telescope with the biggest mirror possible, put it out in space and just look at the closest terrestrial planets to try and look at those atmospheres. I'm not jazzed about that. I'm not excited about it. I'd much rather send more missions to places like Europa and Titan.
My husband told me not to ask questions, so here I am. When you said on Earth the comet like dropped off water, like can you just explain further what you mean
Yeah.
by that and like how it kind of like multiplied into what it is?
Yeah, totally. Yeah. So we believe that the earth that formed, like the core of the earth that formed was dry. It didn't form with water accreted onto it. It was able to survive for long enough for comets to enrich it with ice after the bulk of the earth mass had formed. And so it's multiple comets bombarding the earth at a time that slowly brought more and more water here. So once water's here, that's it. It doesn't like multiply or anything. But we think 100% of the water on Earth was delivered from comets. And we know that from chemistry, from looking at what's called like isotopolog ratios. So we can see like how much of different like types of water are here. And that kind of lines up with the cometary signatures of water as well. I hope that made sense.
I'm going to ask just one question. What are your thoughts on panspermia?
That is what life was like delivered here, right? Via a comet.
For anyone who hasn't heard of it, panspermia is this hypothesis that life may have started somewhere else and life could potentially be seeded on other planets through natural delivery processes like comets or asteroids.
I don't think it's impossible, but I think it's easier to form life here than have it like shipped from one side of the galaxy to the other.
Well, thank you all. We'll just give Jenny one final massive round of applause. Thank you so much. If there's anyone else that does have further questions, feel free to drop me an email and I can pass it on. I can pass it on to Jenny. But for now, I'm going to introduce our second speaker. Dr. Xavier Portillo. Xavier Portillo is a Kavli-Lockean postdoctoral researcher fellow at the Origins of Life Initiative at Harvard. His lab is in the Department of Genetics at Harvard Medical School, where he works closely with Professor George Church. He primarily works on RNA evolution and design for use in outer space industry, medicine, anti-aging, de-extinction, and understanding the origins of life on Earth. He uses biochemistry, molecular biology, and machine learning to understand how RNA structures informs and regulates biological function. He completed his PhD at Yale University and his undergraduate degree from the University of California, Santa Cruz. He enjoys playing guitar, singing, cycling, and surfing in his spare time, which is infinitesimally small at the moment. Javier's title is An RNA World, Past, Present, and Future. Please, enormous round of applause for Dr. Javier.
Thanks, Philippe, for that introduction. And not only did Jenny set the bar really, really high, but also set the bar really high for me to explain how life is formed on Earth. So I would just talk to you about the different theories that we have regarding what we think happened at the origins of life. So I'm just going to pull up my talk really quick. And also all those things that Philippe was saying are not that impressive. It's all the same thing. It's RNA evolution. So I'll show you here in these slides. So my title of my talk is titled An RNA World, the Past, Present, and the Future. And so my research is really, really small. So Jenny is out in outer space in the observable universe. The things that I look at are quite small, but actually the size scale are quite similar. So we'll start off with, I'll call your attention to this size meters here. So this is in meters, 10 to the 27 meters. And we're just going to see where we are relative to all the other things that I'm going to be talking about and also that Jenny was looking at. So here are some, so 10 to the 24 meters. We're looking at some large galaxies here. We're zooming in to parts of Jenny's talk where the pillars of creation are coming up soon. That comes up just right around here, pillars of creation. And we're still moving down the orders of magnitude here. And then we're moving into different suns, and we get to our sun at about 10 to the 8 meters. There it is, that's our sun. And now we start getting into our solar system and the planets in our solar system here, and now other things that we can start to understand or wrap our heads around the size. So this is 10 to the 4 meters, and we're zooming in over to where we are. So we're at 10 to the 0 meters, so we're all about this size here. But the stuff that I look at is really, really ever so small. So we're zooming in to the smallest object that could be seen with the naked eye. So the human ovum is shown here, various different types of cells. My research is here at working with viruses, DNA, RNA. And then so we get down here to a protein alpha helix. The molecules for the prebiotic chemicals would be this scale 10 to the negative 10. And then there's not a lot for a really long time. So here are just the canonical atoms, protons, neutrons, and we keep going all the way down. And so our furthest theorized thing that we can think about is this quantum foam and string theory, which is down in the negative 34, I believe. So there's technically more distance inside of us than there is outward. So we're not even in the middle of the whole orders of magnitude. So here's where we are. So we're at 10 to the 0. And so for us to the sun is about 8 orders of magnitude. And we are in the opposite direction here for nucleic acids. So nucleic acids are about the size of us in comparison to the sun. Subatomic particles like a neutron and protons are about the same scale of a difference. So this is all kind of just like relative to where Jenny's research is over here this way. I'm on this direction here. And so, yeah, as I was mentioning, there's more space if we go inward than there is outward to the observable universe. Of course, there's probably stuff beyond, but as far as we can observe is 10 to the 27 in meters. Whoops. So I wanted to see what these are internet searches for the phrase for the word life, what people are asking for with internet searches. And I was wondering if people were asking the same questions as I was asking for life. So yes, they are asking the exact same questions that I'm asking. Will life ever be affordable again? Why is life so hard? That's a good question. When life gives you Lululemons, that's actually things people are searching for. But then there's these questions that are more pertaining to my research, where it's can life come from non-life, and how life works. Did you have a question? Oh, sorry. Okay. I know it is. So we have an incomplete definition of what life is. So life, depending on the dictionary or the textbook, is defined as an organismic state characterized by capacity for metabolism, growth, reaction to stimuli, and reproduction. Metabolism and reproduction keep coming up on most of the definitions of life, and that disqualifies viruses, even though viruses can pretty much hijack metabolism and are able to reproduce. So we have to properly define what life is to answer the question of how do we find it outside of Earth? So we're not usually looking for a little furry creature on planets, we're looking for signatures for life. So Dr. Lori Barge from Caltech and JPL has this beautiful perspective on redefining life as a spectrum. So she actually works on some of these probes that are being sent out to the different locations that Jenny was talking about. And the buck stops with her. She has to decide what sensors are going on these shuttles, what sensors are we sending out, and what are we looking for. I just want to call out on the left-hand side, I have another little orders of magnitude scale so we can see where we are. So right now we're looking at 10 to the negative 10 to 10 to the negative 7. And so for reference, it's us, an ant, human egg, E. coli, DNA, carbon atom, and they're all 2 orders of magnitude between each one. So I'll just leave that up there as a helpful reminder of what we're looking at. In these prebiotic pools that Jenny was mentioning, we have the famous Miller-Urey experiments from U Chicago and later UC San Diego, where you can get a lot of just prebiotic chemicals, simple chemicals like cyanide and water, and react them in the flask. You do temperature fluctuation, you spark it, and you get a lot of these building blocks. So these are the building blocks that are eventually going to form biologically relevant molecules like RNAs and amino acids and lipids. And then eventually you get to these functional polymers. So my research is here. I'm a biochemist, biophysicist. So I look at secondary structure of RNAs. And so the idea is that at some point you would have a system arise from here that would be, genomes and coding information and creating little machines in the form of enzymes or ribozymes, which are functional RNAs. So where do we draw the line of what biology is, right? So maybe we visit a planet that is not quite in the time in which we would expect canonical life, so cellular life, right? But maybe it has some of these polymers that we can actually probe for. So Dr. Lori Barge is actually one of the persons responsible for saying, okay, we should be looking for some of these prebiotic molecules because there's more time between this and this than there is from the beginning of cellular life to us by many orders of magnitude. And I'll just pull that up in a second. So now we start to look at the same questions that were on that internet search earlier. So how does life work, and can life come from non-life? And so the way in the field that we're trying to grapple with these questions is, number one, the central dogma of biology. I'll bore you all about that later. And then this concept of an RNA world where I did my PhD research on. So here's the boring part. So you all saw this in high school biology, or maybe some of you are not there yet, but you will see it. And it will be the bane of your existence. But the concept is that all life on Earth shares these rules and regulations. So DNA is replicated, and that stores the genomic information that codes for us or single cells. And DNA goes to RNA, specifically messenger RNA, and then the messenger RNA is then used to generate a copy of a protein. But what we've seen is that there's many exceptions to the central dogma of biology. So back when the central dogma was proposed, it was very straightforward. But since then, we have found viruses that code their own genomes. We've found viruses that can do reverse transcription, basically go from RNA genomes to DNA genomes, and many, many exceptions to the central dogma. So then that poses the question, how could the system arise if each one is dependent on one another? And so that's where my research comes in for this RNA world. So the first question is pertaining to life on Earth. If nucleic acids are required to make proteins and proteins are required to make nucleic acids, how can we imagine this system to just spontaneously come up? So there's this chicken and the egg problem. But RNA can serve as both chicken and an egg. So RNA is very similar to DNA. It's just more catalytically active. It's also less stable. And it can store genetic information, and it can catalyze chemical reactions, including this function here called RNA polymerization. That's the ability for one RNA to make another RNA using hereditary information, coded information. So then the question is, can we get a self-replicating version of this RNA? So is RNA capable of self-replicating itself to serve as its own template and also catalyst for evolution? And then eventually leading to the synthesis of proteins and eventually genomes. And I promise I'm going to stick around to answer a lot of your questions, so don't worry. Okay, so here's a timeline for life on Earth. So Jenny was talking about the Big Bang. This is in billions of years ago. We're still looking at this scale here. So the RNA world is a period where life before DNA and proteins. And what I mean when I say proteins, I do mean like large functional proteins, because in the prebiotic pool, you do get amino acids and you do get dipeptides, tripeptides. So you do get some proteins. So when I refer to proteins, I mean functional proteins, large functional ones. When genomes and enzymes were regulated entirely of RNA. So here's the timeline, and we're going to go through each one of these steps through the subsequent slides. So formation of Earth, 4.5 billion years ago. The prebiotic chemistry is about 4 billion years ago. This RNA world is about this, it's proposed to be this amount of time. There's no biological record, so there's no way for us to actually know that this would be the exact time. And then once you have the first DNA protein life, then it rapidly accelerates. So if you're searching for life on another planet, what phase are you searching for? Are you searching for the prebiotic chemicals? Are you searching for these polymers that would be functional polymers that around this time? So we have hidden clues in biology today. So all of the proteins being made inside of you right now and in all other cells are created by this ribosome complex. The ribosome is a mix of proteins and RNA, but you can remove all of its proteins and it still works. So it's a catalytic RNA that makes proteins. So the ribosome communicates with this tRNA to read mRNA. So mRNA is what codes for our proteins. Ribozymes, as mentioned before, are catalytic RNAs. And most people don't think about the energy that's used in your body, but that's the RNA version of it as well. So all the ATP inside of you is an RNA version. And there's so many more RNA functions that are yet to be discovered. RNA continues to serve a major catalytic, regulatory, and information storage role in all of extant biology, and it continues to evolve to this day, usually through random mutagenesis that makes it more fit in the environment that it's found in. So where did this RNA world occur? So there's two theories. The leading theory is the shallow pools. So as Jenny was mentioning earlier, these are like soda lakes, like Yellowstone National Park or Goodenough Lake in Canada. And we also have these deep sea thermal vents like in Marianas Trench. So you have the energy source, you have all of the molecules that would lead to these polymers that are self-organizing to generate single-celled life. And this is the oldest evidence of life on the planet. So these are pictures that we took to a trip. We took a trip to Australia earlier this year to study these stromatolite formations. So these are living rocks. So these rocks are generated by layers and layers and layers of bacterial mats. And this is a fossilized one, but there are living stromatolites in a location called Shark Bay in Western Australia. And the concept here is almost like it's like a New York skyscraper. You have people living or cells living here at the bottom. You have cells living in the bottom middle, all the way up to the very top. And if you cut it down the middle, it looks like this tiramisu here with these layers. These stromatolites are not that old. They're only about 4,000 years old, and the cells inside of them are only a couple days old. But if you drive 8 hours north of Shark Bay, we find these fossilized stromatolites, which were zircon dated to about 3.2 billion years ago. And you can only get these rock formations with biological life. So the first time we went to Australia last year, we had a professor who is working with the Mars missions and studying these geological formations, because when you're looking for life on other planets, this might be your best bet. There are no living cells in these, but if you're able to see something like this, then perhaps there was a phase where life did exist on that planet. So from these locations, so these are mainly prokaryotes, but you do have some eukaryotes here. So this is also a candidate for the origins of a eukaryotic cell, which would eventually lead to the diversification. So we're all kind of connected down here through this last universal common ancestor called Luca. And so from there, you would have the branches of life. And so now we're looking at this timescale here. But as I was mentioning before, we all share this central dogma, this updated central dogma, where RNA is sort of in the middle, where RNA can replicate like viruses and move back and forth and control the production of proteins. So RNA is still playing a major role in life today. So RNA is still running the show. The most recent Nobel Prizes in chemistry and medicine were awarded for RNA machines. One specifically for the earliest immune system that bacteria were using, which is fully guided by an RNA, which is the CRISPR-Cas9 systems. So the CRISPR is the smart part of the system, and the Cas is just a dumb protein that just cuts. Everyone remembers the war between an RNA virus, COVID-19, and the hero mRNA vaccine against said virus. And then the most recent Nobel Prize awarded to a professor in my department for microRNAs, which is just a mini version of these CRISPR systems. So the microRNAs are used to destroy messenger RNAs. So we're still discovering RNA's mysteries billions of years later. So this is my research. So I did my PhD at Yale University with the NASA fellowship, and we're concerned with an RNA molecule here deemed wild type. So this is a functional RNA that can make other RNAs. So it can make tRNAs, it can make other portions of the ribosome, it can make all sorts of different fun RNAs. And the goal is to try to get it to self-replicate. And so over 52 generations of directed evolution, this is in vitro, so we were actually doing this in the lab. You see all of these different enrichment scores here. The darker the blue, it means that one particular variant was highly enriched over generations. But as you hit it with really stringent directed evolution paradigms, you start to enrich for one particular variant that's much better than the others. And so over 52 rounds, we were able to get this novel function on this. Sorry, I have this little fish cartoon to show a gain of function here, which is like growing legs. So we were able to get an RNA that can make its grandparent. So it cannot make itself, but it can make the grandparent. And the grandparent can make it. So we do have a form of self-replication for RNA, but it's a little bit cheating because it's using the ancestral version. So the parent of this RNA is called the class 1 ligase. It's an RNA that can covalently bind to other RNAs. And then these polymerases are able to build RNAs 1 unit by 1 unit by 1 unit. And because it's not 100% fidelity per addition, then you can initiate something like Darwinian described evolution, where the mistakes that it makes could be serving a novel function. So that's my research in my PhD. But what I'm doing now is I'm using these in vitro. So this is what I was doing in my PhD, so evolving the RNAs in vitro. But now I'm evolving RNAs inside of cells as well, specifically mRNAs and other regulatory RNAs called riboswitches. We're also interested in looking at natural selection paradigms, such as those stromatolites, to track the RNAs in those skyscraper communities that I was describing. DNA sequencing has come down in price quite a bit. And so it's quite inexpensive to take multiple generations and get hundreds and hundreds of gigabytes of evolutionary data from these RNA. I developed the bioinformatics pipeline so that we can align sequences and group them together through family clusters. And then we're developing machine learning algorithms to see the minor perturbations in the secondary structure of these RNAs. So we want to see how the secondary structure of this RNA is evolving over time and how we can tie that to its function, its gain of function or its loss of function. So this is the future part of my RNA world talk, is trying to get machine learning to predict the future of these RNAs. So our RNA targets, as mentioned before, are mRNAs, regulatory riboswitches, catalytic RNAs, and also later on down the road, ribosomes. So here are ongoing research directions. We recently have a paper in bioRxiv for using RNAs to combat, so for anti-aging purposes. So you could scan this QR code if you're interested to learn more how we're using RNA to combat these big problems, like in medicine, in organismal de-extinction, of course, answering questions for the RNA world, and we're interested in doing in vitro, in vivo, and also looking at in nature evolution of these RNAs. And so specifically, my work is space biotechnology, understanding the RNA world, and also learning more for medicine. So that's it. That's my whole talk. I want to thank you for listening, and thank you, Philippe and Jenny, for the wonderful setup, and I'm happy to answer any questions.
Thank you, Javi, for an amazing talk. Amazing talk. I will take a couple of questions. I'm going to start off the questions. I don't know if this is actually a question or a comment, but your talk just reminds me of the struggle of detecting extraterrestrial life and how difficult it is. Because my understanding is that we have never detected RNA on an extraterrestrial environment. But the question or comment that I have is, even if we did, if we hypothetically we found RNA on the surface of Mars, like right now, that does not mean it's evidence of life. Is that correct? Yeah. So that's just to highlight how difficult it is finding aliens, right? So more of a comment. But are there any questions? Yeah, right here.
So this may be a dumb question, I don't know, but seeing the chronology of how, the life evolved from the Big Bang to diversification of life, it made me kind of think of future. Are there any predictions or studies? Are there any ways to look into how life is going to continue to change on Earth or wherever else?
Yeah, thanks for that question. So the only thing constant is change, right? So if you pay attention to the selection pressures, you can almost start to see which way the RNA would go. One example through my PhD research is the type of selection pressures we control in the lab. So sometimes we give this big pool of RNAs, it's like 10 to the 17 variants of this RNA. We give it a challenge for like 24 hours to build this little tool. And if it builds the tool, it'll survive. And the variants that don't build the tool are thrown out. And then you amplify the winners. So that's how the experiments go. And so we're controlling the pressure on these things. But what we realize is that the structural, the secondary structure of this RNA was dependent on what sort of tool that RNA had to make. So in one, it had to make like a little hook. In another one, it had to make a pair of scissors. And when it was making the scissors, the secondary structure started forming almost around it. So the selection pressure will inform what direction it's taking. And we didn't expect that. That wasn't something that we thought. We thought we were just going to see it sort of linearly go to a secondary structure. But the minute we gave it a new challenge, it was all bets were off. So it's really exciting stuff. And so it continues to evolve to this day. Yeah.
Any more questions? Yeah, there's one over here.
Are RNAs present in any other worlds that we know of currently?
No, that's a good question, but unfortunately, we have not. We've not actually been measuring. Recently, we have been able to generate really small PCR machines. PCR just amplifies RNA or DNA. We've made really small ones, so some of those have made it onto the International Space Station, but we've still have not been probing for polymers. Nor would I think it'd be responsible to just probe for just RNA, because it could be something else that isn't RNA, like PNA or XNA, which is a similar polymer, but the chemical structure is different. So we would just be shooting in the dark if we were to look for RNA as we understand it today. But thank you for the question.
Any more questions? Any more? Yes.
I have a few questions about your circular RNA, if I can call it that. How long is it? And did I understand correctly that you were able to evolve it to sort of function on its own in a soup and complete this replication cycle numerous times? And if so, how many cycles could you get it to run?
Yeah. So first of all, it's cheating. It's not like the actual replication, and I'll explain it in detail. To answer your first question, it's 183 units long, or 183 nucleotides long. It's single-stranded, but it folds up on itself, and the folding pattern is what makes it catalytic. So the way that the self-replication works is this RNA was evolved by a professor from Harvard who got the Nobel Prize for telomeres, Jack Szostak, who's now at UChicago. So he evolved this RNA from a random pool of RNAs. So not from anything that's living, or not from anything I know that we understand.
Your starting material
was
randomly
random
evolved?
RNAs, yeah. And he picked out the class 1 ligase about 15, 20 years ago now. And so this RNA was barely able to stitch together 2 other RNAs. So it's a ligase. It connects, covalently binds 2 other RNAs. And so since then, there's a lab at Cambridge University in the UK. There's a lab in Canada, and then the lab that I worked at as a collaborator at UCSD at the Salk Institute that continued to evolve that RNA. So they got that RNA to the point where it can build other RNAs 1 unit at a time. So through RNA polymerization, as opposed to the ligase function. So going back to your original question, this polymerase that I was evolving can make its grandparent the class 1 ligase. The class 1 ligase can then ligate together two pieces of the great grandson daughter, and then we have self-replication through that cycle. But the holy grail is for it to make a copy of itself, which you can imagine is extremely difficult. If you give it a template, it just zips up to an RNA helix. And also there is this, it's called the eigenerror threshold. There was a mathematician who calculated what a theoretical polymer would have to have for velocity of polymerization and fidelity. So this is the accuracy of how it builds. We're not there yet. So we need about 98.9% fidelity. We're at about 86% fidelity. So it still makes a lot of mutations when it does this. So we're not at the eigenerror threshold. So velocity, we're still just slightly too slow, but we're almost there. And fidelity, we're not even in the ballpark yet. But happy to talk more afterwards too.
Okay, if there are any further questions or if there are any questions online, please feel free to send those questions to me and I will pass them on to our guest speakers. For now, just please one more round of applause for Javi. Okay, so if anyone missed it, there's a table in the hallway that has packets of lettuce seeds, which you can help yourself. They're there because we grow lettuce in space on board the International Space Station. And lettuce has shown that it can actually grow under microgravity. And so it might actually be a food that we use to when we send our future astronauts to other worlds. So it's pretty exciting. So help yourself to some lettuce. There's also some stickers of tardigrades. Tardigrades are good model organisms for astrobiology because they're little tiny animals that pretty much survive anything. So help yourself to stickers. Right now, what we're going to do is Lisa Bravata and Tom Burns, who are curators of the Harvard Plate Stacks, they have a small exhibition at the back, which I think, Lisa, like 10 minutes, is that right? So it's going to be like 10 minutes, just kind of show and tell. Some of these are really precious, so please avoid touching them for now, unless Lisa gives the green light. And then after that, I'm then going to do a tour of the Great Refractor, which is this gigantic historic telescope that we have here. So that is the plan. So for now, we can go see what Lisa has to show.
Yeah, you guys want to like gather and want to use these shirts, like.
I don't understand how